Welcome back, everyone. So it's on behalf of ISB, it's my honor to present before you the Biocuration Lifetime Achievement Award 2023, which has been awarded to Dr. Sandra Orchard. To just summarize about her contributions to biocuration, she uh, she has been associated with this field for more than 20 years and dedicated uh, his work towards the major repositories which are useful for researchers around the world, like Uniprot, Complex Portal, Enzyme Portal. She also contributed towards Intact, Interpro, and gene ontology. And the impact of her work is quite evident from the publications and also from the proteomics data standards that she contributed towards. Then she also empowered researchers worldwide, and she has been the mentor, advocate, and leader, and inspired next generation of bioeducators. And also, she is the champion of education and community, where uh, 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 she also established the first formal educational qualification in biocuration, and her association with ISB has been long. She also served as treasurer, chair, and also chaired and contributed to numerous sessions of biocuration conferences as well as the committees. And when I asked some of her colleagues, they are just praised her as an incredible mentor, and having her as a support makes them makes them feel truly empowered and confident. And also, they have a suggestion for EBI that EBI should plans to hire 2 people to replace her after her retirement. And Nicole, as she really admired her leadership and contribution towards the society and biocuration. So Charita also praised her for her support, generosity in sharing both the knowledge and her time. And uh, she uh, she said that she's a true super curator. So without the delay, let's we all congratulate Dr. Sandra Orchard and invite him. Thank you.
Jigsaw puzzle. I'll do that instead of giving a talk. Far more fun. Okay, am I on? Yes. Wasn't really given a very clear, well, any remit for this, so I was allowed to talk about what I wanted. So hopefully I'll be summarising a few of the points that have been made by other speakers, bringing them all together and perhaps rounding up and discussing a little bit the future direction of biocuration. So traditionally, and particularly maybe associated with Uniprot, the role of the biocurator has often just been seen as reading papers and summarising the data. So there is a conception by many people that LLMs can also do this. So I have had it said to me that the day of the biocurator is probably coming to an end. I would challenge this, but at the same time, I don't think jobs stay still. They ought to change over time. They ought to grow and meet new challenges. So we do have to reevaluate our role constantly and do so in the light of advances in technology. But the first question we asked at Uniprot when we were faced with this onslaught of LLM mania was just how good are they? Can we trust them? Are they good enough for Swissprot? Or do we regard them as a tool that can help us annotate tremble? hopefully I don't have to explain the difference to this audience, but Swiss Prots are reviewed part of the database where the manual curation is key, and which has always been taken as sort of the gold standard of information on protein function globally for many, many years. And we don't want to lose that reputation. But at the same time, we have always been faced with the 250 million entries hanging over us in tremble, which we don't have the curation capacity to annotate, even if the publications were available. And in most cases, there just isn't any data out there for us to capture anyway. So we've always been dependent on some sort of what we call automatic annotation, which started off as simple annotation transfer, and over the years has become more sophisticated, and machine learning has replaced that as our major tool. But obviously we had a range of opinions of just how good LLMs are, ranging from the Alex Bateman, yay, we can curate everything with these, to our more traditional curators who have much same opinion as the British Luddites did when they first started breaking up the steam-driven looms, in that they just didn't trust them at all. So we really needed to evaluate them to just see how good they were. So we've done a series of tests. I'm just going to focus on one of the very early ones because it was our first attempt to put numbers at this. So we took 20 papers initially. We've built up on this since, but these are the one I've got the data for. And we did a side-by-side comparison, manual curation versus LLM. It was Claude 3 Opus we used for that. And we looked at the precision recall. Well, when the selected model was asked to extract and merge data from multiple papers. So I've said this, the temperature we set to write down at 0 because we wanted consistency, we didn't want creativity. There were a list of 20 papers deliberately that we picked that were deliberately not in the public domain as curated entities already. So they weren't in the gene ontology, they weren't in UniProt. So there was no chance that the LLM would go and find that information themselves. We used the PDF of the entire paper as the import. And again, we made life as simple as possible for the LLM, which chose ones that didn't have masses of supplementary materials to work from. So it was just the paper, and we restricted the prompt to ask for experimental data only. And we use this to calculate the precision recall. So those who have never been exposed to the UniProt flat file format before, this is the, on the right, you can see the work of the curator. And on the left, we fed Claude the slimmed down version of the UniProt manual. We weren't that cruel to give it the whole thing, and asked for it to produce the similar sorts of line types. And on the whole, as we've heard over and over again over the last few days, they did a pretty good job. But there was information missed. Pick that particular paper because it was very clear where the LLM just failed. And this was just doing, at this stage, it was just doing single papers. So we calculated the precision recall by looking at true false positives versus false negatives. And for a single paper, we got pretty impressive statistics for the LLM versus the curator. So it managed about 90% precision and 81% recall. But we did find that the recall in particular dropped quite noticeably when we then asked for multiple papers to be annotated as a single exercise. And we also found that the LLM started skipping papers, just totally ignoring ones. We couldn't explain that. And it didn't seem to be related to the number of papers we fed it. It was just totally random. We couldn't work out what was happening there. So in fact, what we found was that we significantly improved our results by going, first of all, going through them one by one, and then on a second round, asking the LLM to combine the data into a single summary. And that way we didn't suffer from missing data. So what the false positives were caused by, often because it was picking information out from background information that the author had supplied as explanatory notes or speculation at the end of the paper. occasionally it was the Swissprot, Uniprot file format that blew its mind, and I can't blame it for that. It's one of the most difficult things we have to teach curators. So they would just put information in the wrong field, basically, but it's still found it. The more difficult problem to solve was where the curator makes a judgment call. So particularly, for instance, knockout information, if it's not a total knockout, we will tend to not include that in an entry, but that was a very difficult concept to get over to an LLM. So we were getting data from partial knockouts, knock-ins that we were not happy about the controls of. So there were biological decisions that a curator can make with 20 years lab experience behind them that the author hadn't spelt out, so the LLM couldn't pick it up. Classical curator problem, trying to map entities to the species, particularly an issue for the mammalian community, where authors don't like to tell you that they used a mixture of human, rat and mouse, and then wrote the paper all about human. So they tend to not declare that in the methods anyway. So it's hard enough for a human curator to pick that up, let alone an LLM. And of course, the classical hallucination. And similarly with the false negative, similar sorts of problems. So we were fairly impressed by those results, though, having said that, with all the caveats, and certainly feel that it's the technology is applicable to tremble. We've had an NIH supplement where we've been asked to look at variants associated with Alzheimer's disease. And in this case, we combine the LLM with, first of all, doing text mining using the PIR Georgetown University eMind program, which is trained specifically to find abstracts related to Alzheimer's disease and related neurological conditions. And then the LLM pulled out the variants, made a classification as to whether the variant affected the disease or it affected the protein, and made both free text summaries and sentence-based summaries of the information there. And we're now preparing mock-ups, because one of the things we're very, very conscious about, which hasn't been really discussed at this meeting, is how we present this to the user, because we do want to make it very, very clear to the user what is manually curated, what is done by an LLM, and what is done by our other existing summary processes, the machine learning, the annotation transfer, so that the user is under no, we hope, does not confuse any of these different data types. So yes, we can use them as a tool for biocuration, but we do, as it keeps on, people have kept on saying over the last 3 days, human in the loop is essential. There is the role of the biocurator is critical, and it's that knowledge that we hold that an LLM doesn't, that allows us to make these biologically relevant decisions. So this is absolutely critical to high quality data. The prompts can get very long and complicated to the point that you're almost over engineering them, but Uniprot has a complicated format. It makes decisions that are difficult to put into a prompt. How do you tell a prompt to ignore all cell line data without having to then tell it exactly what it's looking for in the way of cell lines, which means you need an infinite list of cell lines for each organism. You can find the data, you can do RAGs, you can improve things, but it does make these prompts very, very unmanageable or difficult to manage. And as I say, we're now doing a lot of usability study on how to present this data. So what else do biocurators do, as well as manually curating, making these fundamental decisions about what information to capture and what to ignore? Well, of course, we're also responsible for providing the high quality data sets that allow for the development, for the training, for the testing of all the machine learning algorithms that are currently under development. We've shown this slide many, many times, but it shows the sort of scale of the problem that Uniprot's dealing with. 250 million sequences plus in Tremble, the 572,000 sequences that are annotated in UniProt, SwissProt are a tiny, tiny tip of our iceberg. And in most cases, our functional annotation is doing a pretty good job of adding additional information to these unannotated sequences. So we can cover about 68% of them using our annotation transfer, machine learning, rule-based systems, and at least give the user some information about this protein. There's another about 20% where we can add mechanistic information, if you like, things like, is there a signal peptide? Is it a transmembrane protein? Does it have particular domains, even if they're of unknown function? We can't necessarily tell you what the protein does, but we can tell you quite a lot about it. But we still then have this remaining 12%, the 31 million, where we just have absolutely nothing. Sometimes we have a submitted name from the author, but that will be it. So we've been working with Google, for the Google DeepMind, for the last couple of years in a joint effort to annotate these proteins. And they're using a T5 model, text-to-text transformer model, to perform a series of tasks where the input is text, the output is text. And we started fairly simple, and our idea was that we would use this technology to name uncharacterized proteins. So we wouldn't add any other information other than a name that we thought captured at least some of the functional information about the protein. So we've managed that quite successfully, we hope anyway. So about 10% of Tremble now, about 28 million proteins have some sort of name. This was an iterative process, so the first set we released were far from perfect. So from the first things we put in was a scoring system so we could just take the highest confidence subset. The other big issue we had was that these programs need vast amounts of data, so we couldn't just give them SwissProt as a training set. It had to train over the whole of UniProt, and author submitted names turned out to be a real problem. Some of them we could quite easily get rid of. They were clone names, so they were alphanumeric strings, KI, AA, 000, 99, whatever. Those were easily filtered out. So were things like protein, which an amazing number of people call submitted protein. So unhelpful names like that we could get rid of, but there were others that caused us real problems because they were names that sounded right, but actually when you looked at the protein, they were very wrong and very misleading. So we've had to build, again with curator input, or curator doing it, series of rules to try and clean these up. And fortunately, one thing that UniProt does have is a very active user community. So as soon as we do something wrong, they don't waste time letting us know about it. And we've, a lot of these edge cases have been identified for us by our community. So the Biocuration input here have been, first of all, putting together the higher quality training set. As I said, eventually it had to train over the whole of Tremble, but we took out a subset to allow for evaluation, and this evaluation had to be done manually, and it is a substantive amount of time. This is something, if you're going to start projects like this, you really have to factor in, because these are manual curators. So I've had to take a person off doing manual curation to spend all her time working with Google doing the evaluation, because there's round after round of data generation. It's not just one little Excel sheet you have to work through. It's week after week you're being given more data to look at. Machine learning may save us time in the long term, but in the short term, it is something you do have to factor in if you're working to deadlines or output. You expect a certain amount of throughput. Proton LM2 is now well underway, so we're now moving to beyond protein names and looking at other fields we can predict. So can we build function statements that look like UniProt function statements? So again, we're using our very unique line type, catalytic activity statements, subset the locations, what keywords can it add, what family and domain information, Google DeepMind are also working with Alex Bateman on PFAM to predict new protein families. So can we bring some of that newer data in? So we are building so again, we have the training set is practically the whole of Uniprot. We obviously keep a subset out for testing. And our test set is quite biased towards Swissprot proteins because we got the good annotation there we can compare with. We've built two tools, one at Google, one at the EBI, for looking at the data once it's generated and saying, does it look sensible? As much as you can for 240 million proteins at any one time. So the evidence that Google DeepMind have built does a lot of alignment work, both sequence and structural, to see whether the proteins align sensibly and fall into groups of proteins you'd expect to create cause. I'm not going to say families because they're bigger than that, but that you would expect to group together. And at the EBI, we're looking at the information that's already in similar entries, doing our own methods for identifying those, and again saying, does this annotation look sensible for these proteins? And then that's at scale, and we're still going through this manual evaluation by a curator using their background knowledge, their experience, and tools such as BLAST and FoldSeek to see whether they agree with the prediction being made by the machine learning. So we've got, say, we've rerun the names with the improvements that we've built in there, but we're also now adding function, EC numbers, some gene ontology terms, and several other. So we've restricted it to that to start with. Function encompasses catalytic activity as well for enzymes. And for nearly 94% of these predictions, they've got evidencer. So this is this Google DeepMind tool, evidencer support from Google DeepMind. The remaining 7.1, we've no reason to think to believe they're wrong. Well, most of them, we've no reason to believe they're wrong. There just isn't any evidence either from Google or from our own systems. But we think at the moment that the error rate is below 1.5%. And again, a lot of this is driven by the submitter filling things in wrong way when the original depositions made to ISND to Embl GenBank ENA, rather than necessarily caused by the machine learning. So we're filling the annotation gaps in Uniprot Tremble. That's our sort of immediate goal. And we're going to put 10,000 out, which we have seeded very heavily to be 10,000 proteins that people are likely to look at. So model organisms being an obvious example, in the hope that they will feed back every time they find a mistake. And then if that pilot is accepted by our user community, then we'll release the remaining 230 million. So we've got lots of new information for uncharacterized proteins, and we can also use this as a code. We can run this over Swissprot as well and say this information is missing in Swissprot proteins, and believe me, there's a lot of work going to be generated from that. And the intention for the MLM3, the follow-up project, which presumably will be next year at the earliest, is to make potentially another tab so that you can see the Uniprot entry and then click on the tab and see with full page predictions. So we'll effectively be creating an entire Uniprot entry just starting with the sequence. So switching gear a bit to protein complexes, there's another example of where we're using machine learning to do predictions. So those who've never heard of it and didn't see the poster last night, it's an encyclopedia resource of macromolecular complexes. So it's reference entities. They've all got proteins in, but quite a few of them also have other molecules, small molecule cofactors, for instance, or long non-coding RNAs in several examples. We've got 20 complexes from 28 species, but we're focusing most heavily on model organisms. And we've nearly 2,500 human complexes already done. SGD, Flybase and POMBase are helping with this. And we've got, at the moment, we think, we're saying full set for E. coli and Saccharomyces, but having said that, obviously new complexes pop up in the literature every now and then and hopefully get captured. And we're working to say coverage for the other species. And you get the what's the topology, the stoichiometry, obviously links out to reference databases, UniProt, KEBI, and RNA Central mainly. We reannotate the gene ontology at the complex level, because you can't just pull in what you get for the protein, as many of these proteins do other things under other conditions, and cross-references, crystals, AM data, etc. So in the last year, a couple of years, we've been working with Kevin Drew, who used to be Ed Marcotti's postdoc, and working to produce HUMAP 3.0, which is predicted complex set from human set from the HUMAP from mass spec experiments, so affinity purification, co-fractionation on columns, and this time round they've also included biotin labeling as evidence of proteins being in close proximity. They build a network from this, they cluster, and using machine learning algorithms, look at those clusters and decide which ones are they believe to be stable protein complexes or not. They use the complexes, the manually curated set in the complex portal as their training and test set, and predicted about 15,000 new complexes, which we've integrated into the complex portal. And independently Triadica's group, UCSD, I think it is, and Emma Lundberg with her co-localization data have been doing a similar exercise, but they produce less, much bigger, less complexes, but of much larger size, and those have also been imported. We weren't actively involved in the generation of those. We've just taken the results. So these complexes, at the moment, we have no information about them at all, apart from a list of protein components. But one of the things we have managed to do is work as part of the same grant, is to work with Gail Kostacker at the University of Edinburgh. And he's runs Proteome HD, which looks at co-regulation of any two proteins across many, many proteomics data sets kept in the repositories. And the idea is that if your proteins are all in a complex, they ought to be co-regulated. So they all upregulated at the same time, or they're all downregulated, depending on conditions. So we can start looking at which of these complexes look to be more biologically sensible than others. And similarly, we can take the data in intact, which is all the IMEX data, and look for those for protein pairs within the complex for which we've already got experimental data. So we can start hopefully separating out those that are real complexes to those that are machine learning over predictions. Again, heavy input from curators. This work would have been impossible without the manual curation of the complex portal, and that contributed to all the data sets in IMEX. Further work supporting the development of a machine learning algorithm. So UniProt tried their own community challenge in 2022, predicting metal binding sites across proteins in tremble. We didn't have all the access to the machine learning community that EDO has, so we weren't as successful, anything like as successful in recruiting people to take part in this. So it was an interesting experience, but the results were disappointing. We only had 6 entrants, and to be honest, none of them did as well as our own machine learning that we developed in-house, so we didn't get very much from it. But, we learned a lot, but it was largely what not to do next time. But as you heard yesterday, we've very successfully worked with EDO for since CAPA 2, in that we've provided the gene ontology data sets that's been the training and test sets for this challenge. And as I've just flipped one of the part of the information from one of the slides that EDO showed yesterday, to show there has been a slow but steady improvement with the big jump that we saw for CAFA5, where we had the Kaggle group behind us. So these machine learning algorithms are improving. And the important thing about these challenges is that those who are top of the leaderboard in Kaggle terms or take part in the previous CAFA competitions have to make their code public. So we can take that code and you build it into our own pipeline. So there is definitely a benefit for us to doing this. But again, it's all based on the manual curation work that provides these test and training sets. This is equally important, not only for the manually curated single sets, but also for curation of large-scale data sets. So it was alluded to on those slides earlier. I've been involved with the Proteomics Standards Initiative for many years. The mass spec data side of that obviously is very large-scale data sets that are deposited in resources such as PRIDE and Peptide Atlas. To start with, this community had to be educated into data deposition. So the focus was more on getting them to deposit data than worrying too much about it being well annotated. So there's always been a certain amount of information gone in there, but as we're now hearing from the people who are trying to reuse these spectra and reanalyze them, not enough. So I did find out what SDRF stood for. Sorry, I couldn't remember when I was asked yesterday. It's the sample and data relationship format, which was, I believe, originally developed by the RNA-seq group, but it's been adopted for proteomics, which encourages, or mandates, hopefully eventually, submitters to add the essential metadata that allows this to be reused. One of the simplest things with proteomics is that multiple files get deposited. So if 50 files have been deposited, you need to know that 1 to 25 is the control data and 26 to 50 hold the test data, the sick person data or whatever they've been looking at. Otherwise, there isn't an awful lot of point in reanalyzing the data. This information is almost entirely missing. And I was at the PSI meeting last week, which is why I couldn't make the weekend workshops, where there was a lot of people trying to do reanalysis who had been complaining for the last 2 years that PRIDE and Peptide Atlas had done a poor job and hadn't got this data added, or that their curators weren't going back and adding it. So we actually, wasn't reading it, but I did enjoy the experience. All these people were sat down and given a large-scale experiment and the paper and told to find the information in the paper. And I had a very enjoyable afternoon going around going, welcome to our world. And now you know how biocurators feel as they sat there complaining about the fact that this information was just missing from the papers. And the sort of quote from the organizer at the end, without well annotated metadata, everything we do is a waste of time and money. If you can't, if that doesn't sum up the value of Biocurator, what does? So another role that we play, and so this is the leading into it, obviously, and again, this has come up over and over again the last 3 days, we create standards, we create formats, and we create ontologies to bring order from the chaos of experimental data. And if they don't exist, we create them, but hopefully if they do exist, we work with them and we add to them. So the HUPO PSI, just to sort of talk about the work I've been directly involved with, it was created back in 2002, so it was one of the earlier standards bodies, and it was effectively the Bioinformatics Committee of the newly formed Human Proteome Organization. And don't pay too much attention to the human, it's the only proteome organization out there. It was just sort of riding on the back of Hugo with its name. So we were given the task of creating, of enabling the creation of public domain data standards that would allow the building of their large-scale repositories, because up until this point, there really weren't any repositories for proteomics data. Peptide Atlas was in its very nascent infancy. And PRIDE was still a sketch on somebody's back of somebody's envelope. MS data, mass spec data was only available in vendor-specific formats, so that was the mass spec group was one of the first to set up. And the other group that started immediately were the protein interaction people that I was involved with. The PSI has grown hugely since then. Other groups have come and gone really as problems have been solved. We've currently got the disordered protein people affiliated with us. Simply the glycoproteins are the most recent group to join. And so we tackle problems as they come along, and sometimes the work groups stay for the long term, as the mass spec group and molecular interaction groups have. Others come and go as problems are solved and are no longer an issue. So we all produce the minimum requirements documents we heard about a few minutes ago. So for molecular interactions, that was mimics. So we all have our own little shorthand for these. Data exchange formats. Initially, they were all XML, but technology's moved on. So nowadays, JSONs are favored for this. The interaction data has different flavors depending on what you're trying to, information you're trying to exchange or move around. And we all create ontologies. I prefer to call the molecular interaction one a control vocabulary, because you really can't do reasoning over it. It is just a control vocabulary for marking up the different aspects of an interaction experiment. We publish it obviously on these, and if you get the chance to join standards bodies, one thing I will say is it does great things for your H factor or whatever measure you use. These papers are well cited, and they're the gift that keeps on giving. So they'll carry on being cited for years and years and years, whereas an experimental paper will have its moment in the sun and then disappear. Whereas standards papers, you carry on getting the citation. And out of this all grew the IMEX consortium, a group of databases that share curation efforts, using agreed common rules. They use the same ontologies, they use the same format standards. Originally, we worked by data exchange, but that's the main issue is that protein interactions aren't particularly well funded. So databases were struggling, losing development, not being able to keep software going. And so over time, everything's ended up in a single database, and we have a common editorial tool that all the different groups can use. And this has provided a mechanism by which the small resources can keep going, because they don't need to run their own database locally. They can just use the web services and put a lightweight website on top of the data that they're interested in showing. And if they disappear, if funding gets cut completely for a year or two, it doesn't matter. The database is kept and maintained and kept refreshed. They can keep their website going, or they can take it down depending on what they want to do. But if in a couple of years' time they get more funding, they can come back and the information's there. So it's managed small groups to keep going with just a curator or half a curator, if that's all they've got down to. And it's allowed them to, when the resources disappeared completely, as they occasionally sadly do, the data hasn't been lost, it's been kept. So all that expensive curation work is not wasted. And I just thought I'd show a pretty picture of Kalpana's poster. I mean, this is a lot of work behind this, which you can't really appreciate from the picture, but it's the work of at least 4 groups have contributed to this. And the IMEX model is very, very detailed. So within this is binding domain data. All the pink lines represent mutations, experimentally induced point mutations, which affect an interaction. And we use control vocabulary terms to describe how, whether it disrupts it, decreases it, or even causes it potentially. And these can then be mapped to disease variants. And because we've used standard formats, they can be brought into the UniProt records, and we can align. If you've got very good eyesight, there's a yellow line there, and I emphasize the blob. So these are all the variants that have been brought in from all the different variant databases using the standard formats, because they all have standard formats. And we can align this to the mutant data from intact, and this potentially pathogenic, this variant which is believed to be pathogenic, but nobody had any idea what it did or why it was pathogenic. We can now see it because it disrupts an interaction or a small, maybe a small interaction network. and you can go into Intact and start generating an understanding of why that variant is pathogenic. And without the data, every data being in standard formats, Uniprot just couldn't do this integration. They're bringing all this information together because we're merging protein standards with genomic standards. It's bringing a whole, different worlds into a single resource. You can also do reanalysis if you've got the proper information, as I alluded to earlier. So one of the projects we've got running at the moment is the PTM Exchange Consortium, which is taking data sets from proteomics databases, particularly PRIDE and Peptide Atlas in Seattle. The ones that have been properly annotated, so we know that they're enriched for PTMs. There are various ways they're doing this, I'm not going to go into now, but we're reanalyzing the data, so doing consistent reanalysis of the data, running this through, and it's allowing us to use mass spec to identify sites of PTMs and what that particular PTM change is. We can do this, we can do a statistical analysis of how confident we are in this, which again, I'm not going into, but we've adopted the bronze, silver, gold traffic light system, if you like, to say the ones that we've got the highest confidence for, the ones that are less low confidence. This is all open source, if any of the model organism databases. So you can see we've got things like some Saccharomyces data, mouse data available, some zebrafish already. Sorry, no, misread that, maize data. And this is an ongoing project, so this list will only grow. And we've added them now into Uniprot, again using standard formats, because this data was well annotated by Biocurator when it went in. And we can now add these into the Uniprot entries and tag them as coming from large scale so that the users are understand where it comes from. We've got the bronze, silver and gold showing there, and we can map them to the underlying sequence. And Biocurator is another thing we do all the time, enable novel scientific research, and we've heard much of these. AlphaFold, would that have happened without PDB and Uniprot? We heard from Paul about the work his team have been doing based on the work of the gene ontology, and there's a paper coming out again. So all of these are Nature papers. I mean, these are high impact papers that we're doing. So this one's actually just been accepted by Nature using tissue-specific protein interaction networks, which is the work of the ICMEX consortium, amongst many other data types. And there are research projects sitting there waiting to be picked up. So now that Paul's sorted out the evolution of proteins, perhaps you'd like to start thinking about protein complexes, because we're building up nice sets across many of the model organisms. These are growing, numbers are growing all the time. So perhaps you can tell me why Drosophila manages to be more like human despite the fact it's only got 2/3 the genome, whereas worm goes off and does completely different things when it comes to protein complexes, despite the fact it's got a much similar number of protein coding genes to human. So I certainly think there's a couple of research projects hidden in that data set, and all of this has been, this couldn't have been, this is the first time it's all been brought together, or is being brought together in a single resource to enable this. And of course, I'm not going to touch on this because it's just part of our work we do all the time, day in, day out. We all do masses of training, outreach, and we also do usability testing, which is often gets forgotten about. But we need to make our resources usable by people. So I'll just, yes, I know I'm running out down to my last 10 minutes. So I'll just, last few slides, where are we going in the future? So hopefully the last 3 days have convinced you all that biocuration will remain absolutely essential for getting the information into databases in a way that makes it usable, reusable by the scientific community. We've got many useful tools out there already, and LLMs are just adding to that number, but they are still tools and they still need that human intervention to make them sure they're doing the job that we want them to. But, and it is a big but, we're going to have to keep on justifying our existence. We went through this in the tech, those of you who have been doing biocuration for years will remember we were all going to be replaced by text mining 10 years ago, 15 years ago. Well, look how well that worked. But we're constantly having to fight against this conception that we don't really do anything useful and we're just there to make grants more expensive. So we have to justify our existence and remind funders in particular. It's not usually the grant. Usually when we get grants assessed, we'll hear that Biocuration, we get good reviews. We just don't get funded for any project that is Biocuration driven. And it is going to be, let me drag the elephant back into the room, it is going to be an increasing issue from large number of resources over the next year or two. So we have to keep on banging our drum at every possible opportunity. Consortia have proven one mechanism for making more or less. So I mentioned several: gene ontology, IMEX, Uniprot is itself a consortia. The Alliance for Genome Resources is another one. It's got pros that you can create central databases and tools that can maintain the data, so it reduces the number of developers, for instance, that you need. Sorry if I've just made half the room redundant. We've heard about the issues of data security, but I would argue that every release can be very easily dumped somewhere else, so we've always got backup. It also cuts the more consortia and the more interaction we have between the existing consortia, we can cut down redundant curation, because we are guilty of Flybase and Uniprot annotating the same paper and effectively pulling out the same information. We know we are, and we should be able to do that better. It's a mechanism for protecting data when a single resource fails. And it doesn't stop the individual resources presenting the data in their own way, should they choose to. We've got APIs, we've got all the data in a properly maintained database. So there are many different ways it can be put out in multiple websites to show different views. But the collaborative curation workshop on Saturday concentrated very much on training. I would suggest in the future maybe you need to broaden the thought a little bit and start thinking about how we manage the curation itself. The biggest barrier to this is always our way is best. We're happy to collaborate as long as we do it our way. It's probably, the biggest hurdle we have to get over. And also funding mechanisms, which tend to be very parochial. So it's actually quite difficult to get a global consortium funded because everyone thinks that's another country's problem. So again, that's a mindset we have to tackle. LLMs, another opportunity, but they are expensive to run and computationally expensive and don't do our green credentials any great thing. And again, I think we're going to be guilty of doing this redundantly. So maybe we can find some way of, once we've run one, can we put it somewhere that another database can access and decide whether this is paper to, worth looking at. And perhaps this is a role that EuroPMC PubMed could potentially play. Just throw that out there. That we could deposit our LLM output with them, and then it can be accessed by other resources. But basically, be proud of what you do. It has an incredible impact, and if you don't believe that after the last 3 days, then there's nothing more we can say, really. But have confidence in your work and take every opportunity to tell everyone just what a great job we do. Yes, your job's going to change. Of course, it's going to change. Everyone's does over time. If it doesn't, you've probably got bigger problems than an LLM coming up behind you. Adapt and make the best of use of what's available to us. Educate the wet lab scientists. They're the ones sitting on the grant panel, and support the work of the ISB. But don't leave everything to them. Outreach is everyone's responsibility, and we all ought to be doing it. So I didn't bother with an acknowledgement slide because when I started writing it up, I realised over 24 years I've worked out with so many people that I was going to need several. So thank you, everyone, and whoever works with. Those that haven't heard, I'm retiring at the end of October. So if anyone would like to come onto the Europe, where the landscape's a little less politically fraught at the moment, the job's up, so please apply for it. Okay, any questions? If I have time. 4 minutes.
We have 4 minutes for the questions.
Okay, I'll start because I've got a mic. So again, thank you, Sandra. That was really an excellent talk and a really great reminder of why it is that you deserve this award. So, so again, thank you. I want to ask a policy question. So we've gotten to the point where we can do these huge analyses. We can, you know, say that some of the output is appropriate to present to users. We can tag it with appropriate evidence codes. We can put a big neon sign around it saying it was generated by AI, klaxons, whatever you want. And then there are a couple of things that could happen in the next, say, 6 months or so. A curator could pick up on one of these annotations, investigate it, affirm it, refute it, refine it, generate 6 more in that area. Or, since it's such a fast-moving field, you know, some shiny new AI engine could come along and you want to run a new analysis. So my question is, what do you do with the old analysis? Do you keep that around? Do you get rid of it? Do you put it in some sort of archived place for historical purposes?
Yeah, I'm not sure about, well, we do have an archive, so we can train all the Uniprot annotations past 40 odd years and right back to the Swissprot Tremble days. I think it's worth keeping, if only to learn from. So all these sorts of things are iterative, and you learn from the mistakes you made the first time round. Yeah, I guess there has to be a cut-off eventually where it just becomes pure history, but certainly in the short term, I think it's probably worth keeping some sort of record of what was added. The traceability, and also, of course, people will have used your results in their analyses. So if you've changed things, this is the reason we started the Uniprot archive, because people were doing proteomics experiments, for instance, on a particular version of Uniprot. Things changed in the year between doing the analysis and publishing the paper. So they had to be able to say, we ran the analysis on this particular set of data. So similarly, when they're using machine learning information in their paper, they need to be able to say, and it was generated from Uniprot version 99 or whatever. So from that point of view, it's important that we keep some sort of record. Keeping a record of the algorithms themselves is probably more difficult, particularly when we're working with people like Google, where that's a bit out of our hands. But yeah, I think archiving does play a role. I think it is important. But yeah, I agree with you. I have to say, I do think there is a cut-off. So you have to sort of make some decisions of what is sensible to keep.
Okay, thank you.
We can have a few more questions.
Oh, thanks, Sandra. Always great to hear you talk. So first, yeah, I can answer your question about why worm is more different from human than fly. It's about evolutionary divergence. They've diverged further from the common ancestor, but we'll yeah, I'd love to talk to you about this one. So, but I actually have a question for you, which is so I think what is often underappreciated, just because it's sort of considered an old problem, is this problem that you talked about user submitted names. So you know what you know, I just wanted to talk first to point out, you know, this is really impactful and a very impactful use of the curator time that it took to get the LLM to the point where you could get reliable names for these submitted proteins. Because I think you know, you know very well, and maybe everybody here knows, that especially for the microbial community, I feel that their trust in sequence databases from this problem of submitted names, right? Because you know these are what users submit with their sequences. There's no incentive for them to curate these names. Some of them do a decent job of it, but on average, you know, you just don't expect it to be good because it's not their mandate. So what I wanted to ask is, I think this is a really big advance, and it's going to make a big difference to the microbial, you know, the community of scientists studying microbes. How are you going to get the word out of this, what I think is a really revolutionary change in Uniprot, and you know, basically, how do you get the word out, and how do you interact with that community from here?
Well, it's not a community I personally have much in the way of contact with. So, but I mean, one possibility is through the genome annotation meeting that's happening in Cambridge, or through that group. Google, in a lot of ways, it's been a bit of an eye opener for us, because we're used to publishing and working with a commercial group who are an academic subgroup of a commercial group who have job security and aren't pushed by quite the same. You need to get something on your CV because you might be looking for a job in a year or two. They don't have the same push to get things written up. They're actually quite frustrating to work with, because we've got at least 2 good papers worth of data there, and which they're doing nothing with, and we can't move without them. So it is a bit of a frustration for us from that way. So the obvious first pass of write it a paper and push the paper isn't really open to us at the moment. So yeah, I think we're going to have to do it through community outreach, identifying the right communities, getting the word out, and pointing out that we are doing this. At the moment, we say with protein NLN1, we limited it to anything that was just literally had uncharacterized protein in the name. We're going to be more confident with the second release and start changing names where there's something in there, but it's just wrong. And that's the point. I think we'll start shouting about it.
So let's thank Dr. Sandra Orchard with a actually time is up. Maybe, yeah, we can discuss during the lunch time. So now for the second session, now I invite Dr. Cochran for his talk.
Great. So thank you very much. So thanks to the organisers for the opportunity to speak. And it's always a pleasure to come to these Biocuration meetings. So great to be here. So if you are if you're in the audience, if you're at this meeting, you're a biocurator, or you're involved in some way in biocuration, then you necessarily spend a lot of your time working in, or certainly around, life science and biomedical data resources. And so this is a set of data resources that are typically very open: databases, services on those databases, tools, standards, ontologies, and so on. A set of data resources that exist not in a single place, but as a highly distributed ecosystem of different activities around the world. And it's become very much a foundation for life science and biomedical research, very much actually, as we've heard at various points, sort of taken for granted by the people that really depend on it. So the reason you do the biocuration work is because you know that it's essential that well-structured, well-annotated, well-documented data are critical for life science and for biomedicine to advance. There's an absolute dependency there. And as a result, you know, society is to benefit from science further along the chain in health, in food security, in biotechnology, and so on. In response to climate change, then of course this needs to continue to happen. So this is sort of pretty well understood, and so I'm going to say a little bit about the Global Biodata Coalition in a second. But we're an organisation that we concern ourselves with the sustainability of biological data resources, and we all know that at any one time there is always uncertainty about the funding for a particular database or a particular activity around a database. And it's been a history of decades of really struggling to get sufficient funding and funding in the right shape to be able to continue to do the things that are really so important. And of course, at the moment, there's an increased level of uncertainty. So some of the really critical databases in this system are really at risk. Things are very fragile. It's comparatively easy to justify this foundational infrastructure for science. But if you go to a funding organization or any other body that you want to invest in it, then like any sensible investor, what they say is, well, okay, yeah, it sounds very important, but what exactly is it? They want to understand what it is they're investing in. And so actually, although we know there are thousands of databases, we have a program to understand the biggest, most connected, perhaps the more generalist databases, and to discover the other databases, and I'll come on to both of those. We actually don't know very much about the system. We don't know who runs it. We don't know who funds it. We don't know about all of the interconnections and all the interdependencies. So what I'm going to talk about just briefly is really just to give you a glimpse into some work in progress to try to characterize this this landscape somewhat. But I'll start with a couple of slides just to introduce or to reintroduce GBC for those not familiar. So the Global Biodata Coalition is a coalition of life science and biomedical and biodiversity funding organizations of different types that come together to recognize that data resources, data infrastructure is really important for their research programs. They recognize there is great fragility, there are problems, there isn't sustainable and well-coordinated funding around the world to support these operations, and they commit to trying to find solutions together. So at the moment, we have 11 funding organizations from around the world. Typically, they're national agencies, they're linked to government. We have a couple of foundations in there. At any one point, we have a number of observing organizations who are, we hope, on a path to full membership of the GBC. But of course, this is only although there's a good representation and some very engaged funding organizations, of course, this is only a tiny proportion of the total number that exists around the world that really need to engage with this. This is a global challenge that needs a lot of input from a lot of different bodies that are concerned about life science and biomedical research. So we work in lots of different ways. We have just recently released a white paper that's taking many different views on sustainability and offering some principles that we should follow when thinking about building a coordinated system for sustaining essential life science data infrastructure, and it offers some models through which that might be explored. We publish various impact studies. We have a current open letter campaign, so we do encourage people to come in and have a look to see what we're doing and sign up to that. And we have at any one time a number of other campaigns ongoing. We operate a group of a forum and a selection process for what we call global core biodata resources. So these are data resources that are particularly connected in this ecosystem. They're feeding data, they're knowledge bases that are generally used, so they're particularly connected and particularly important in the overall infrastructure. We operate various workshops and sessions, so we co-organized an AI session, for example, on Sunday here. And we have various activities around engaging funders and various biodata resources. And we have with us some hard copies of our recent highlights document. If you want to get a quick 30-minute glimpse of what we're working on, then that's a good starting point. So onto the trying to understand the landscape somewhat. So we're using three sources of information or three sources of data to explore. The first is the Global Core Biodata Resource Program that I mentioned. So this is a set of data resources, currently 52, that submitted applications to a process either in 2022 or 23, and there will be future rounds as well, saying they went through an external expert review against a set of criteria that define what a global core biodata resource is. And as part of the application process and as part of the ongoing communication, we know we can characterize and ask questions. We know a lot about these data resources. The second source of information is from a piece of an analytical workflow that's sort of literature based. It's based on the premise that if you are running a database in the life sciences and you want people who you don't yet know to come and use that database, so your database is somewhat open, then you write a paper about it in the scholarly literature and people will know about it and they'll come and use it. And that's a paradigm that's used by many of the databases. It's not used by all, but that way we know we can pick up a lot of the data resources that we wouldn't otherwise know. So we have a text-based approach. We look through literature, we find those publications, we publish the methodology for this. It's an interesting natural language method, but we have high confidence that we're finding those publications not just mention a data resource, but those publications actually describe a data resource by the people who run, authored by the people who run the data resource. So at the moment, we find almost 4,000 different data resources that we can identify through that route, and we can pull out various bits of information about funding, about people, about where and when and what kind of data and so on. Then the third source is looking at citations as yet in the scholarly literature so far, and looking to see where people are citing accessions and identifiers from different data resources, and we're focusing on the global core biodata resource set. So we're looking to see where people are citing a particular data record in their publication, and for us that's an expression that they are using that for something. So that's been a useful record for some reason or other. So using these three bits of sources of information, we then build it into a single schema. So the information comes from a different source and it's quite different, but what we can begin to do is look at connecting things about the data resource itself with things about the funding, with things about the publications, so there's access into onward citation networks and so on. And so we can build all this together and ask lots of questions. And we're just really starting this process. And our priority is to help us to engage funding organizations and engage people who run data resources. So it's already led to a few things that are that are useful and interesting. So to start with, so we ran the inventory. So this is the mining to find data resources that publish themselves. We found a growth between 2022 when we first ran it in 2023, and we'll run it again quite soon. We're running it again. So the numbers seem to be increasing, and we're not changing the method. So perhaps it's republication, perhaps it's new databases being born. We're not sure, but we'll look into that. We find reference to over 17,000 different entities that are funding database activities, and that's extraordinary. You have to exclude a lot of those because they're likely to be very internal institutional programs or projects, and not particularly substantial. But in there is a wealth of funders who are funding databasing and data resource activities. So we also have a list of people. So we've started to work through the list, so prioritizing those that are most highly cited. When we've now got to a few hundred, we're sort of hearing back from many of a few hundred of the authors of the papers who are describing the databases, so the people that run the databases. So we can begin to build a community of the people who so far don't necessarily, in fact, many don't, we know from the responses, they don't know about the things that we're doing. And then looking at the citation of the data records, we see that 17%, if you take the life science and biomedical corpus of scholarly literature, 17% of the publications have they cite an accession number, at least one accession number from a global core biodata resource. And we know we don't find most of them. We're hugely undersampling. We haven't even looked for it. We can't even yet look for all of the GCBRs, all of the data resources in there. So this is an underestimate, but that's a huge proportion. If you just cut down to the last 10 years, and we think that the discovery of accessions and the citation of accessions was less practical more than 10 years ago, that number rises to 29%. So almost a third of the scientific publications in the domain are using a data resource in some way, and we have evidence for that. So I think that's particularly compelling. So if you care about life science research and you're a funder that runs a life science research program, or you're a government, a nation that cares about life science research, then this is a very clear demonstration that actually these data resources are really important for that to continue. Another interesting thing is that if you look at the map of where the data resources are, so where the citations of records within data resources are coming from, then every country, or with the exception of one or two, everywhere in the world, people are using they're writing papers that are citing records from the GCDRs, from the core data resources. And when you normalize, I haven't done it here, but when you normalize for the total number of publications. So you try to find a proxy for the scale of scientific activity within the country you're looking at. It's actually very, very balanced. There's a very narrow band. That ratio is very narrow. And so wherever you are in the world, if you're doing scientific research, then you are using to this similar extent these global core biodata resources. So there's some compelling stories that will continue to develop from there. So some of the things that we can start to do. So this is Sandra showed a really nice network diagram. This is what in my experience, most network diagrams look like before you start. So essentially what we're doing here is we're just linking countries together, but we're linking them based on the co-authorship of the publications that are describing data resources. So it's just sort of at one level. So these are the people are working together from these countries to run data resources. And of course, it takes a long time to understand this. And we do, we have distilled it down a little bit. And so, for example, if you care about India, then you can see that scientists working in Indian institutions have co-authored papers that describe databases with scientists working in institutions in many other countries. And this is really important. If you go and talk to different countries, they want to understandably, exactly how things are happening in their country and how collaborative and how international things are from their perspective and their scientists' perspective. Then you can also begin to define different roles. So here we have a little bit more sophistication. So Denmark is a little bit more sophistication of the analysis. So Denmark is a host organization for a data resource. The United Kingdom is a funder. Canada is also a funder on that same data resource. There's an affiliation, so someone in the United States is also working on that data resource. So we can begin to drill down. And I won't show it here, but we have scientific domain and specific areas of data types and so on. So there's a lot of understanding we can get of this landscape based on these kind of analyses. So the kind of thing, and I'll just finish with a list of priorities for us. So we want to use this to engage more of the people who aren't yet talking to us and know about what we're trying to achieve from the data resource provider community. More of the funding organizations around the world who care about this or should care about this, or we will argue should care about this. We'll break things down into functional classification because we're working with the literature. We have things like mesh terms, and we have all sorts of other mined outputs that we can use to understand the domain of science and exactly where these data resources are working. We can look at the secondary and the tertiary citations. So the first thing is someone writes a paper, then someone else will come along and cite that paper saying, we found this database really useful. So then there are onward networks, which will give a lot of power and a lot of statistical evidence for how impactful these things are. We can look at other corpora. So the patent literature, for example, so far we're just working with the scholarly literature, but the patent literature is another area where data records are cited. We can look at the citations. So many of the data resources, particularly the bigger ones, track large numbers of publications that are associated with their records. So the map I showed you is based on publications where we find an accession. So this is the reverse. This is where not quite the reverse, but this is where the data resource itself knows about a publication. So that will be overlapping, but it will add more data points into the system, into the analysis. Lots of relationship network analysis, and then really all of this to profile funding sources and to understand the models that are used in funding. And so with that, I will close. We have a newsletter. Do have a look at the website. Chuck and David are here, and they have, and I have some of the brochures, the 2024 highlights. And do have a look at the white paper. It's a long read, but it's an interesting read, and we invite people's comments. It'll be great to see further comment back to that. And do have a look at the open letter, and if you can, lend your personal or your institutional support for what we're trying to achieve. And with that, I close. Thank you very much.
So we have 3 minutes for questions.
Thank you very much. This is admittedly a silly question. On slide number 7, you showed the map of where the citations came from. And it looked like, yeah, it looked like a number of them are coming from, those dots are located not on land masses. Are we looking at research being done on
This is
ships?
no. This is a this is yeah. This is a dictionary ontology issue. So what we're doing, the country list we're using to parse out where things are from is coming from a country list that includes, it treats not at, so if there's an overseas territory, it treats that as a separate. So some of the islands, some of these dots in the oceans are islands. Even though they may be overseas territories, this makes them look like they're separate nations, they're not. So there's a sort of demerging there. We also have a number of seas that are mentioned. So we're borrowing the dictionary of countries from a particular place. We need to refine it. So you see some dots where you wouldn't expect there to be a country. But all the countries that I think with the exception, I think there's an exception in North Korea, but apart from that, there's at least a dot in every country that is a country of the 190 something. Is that, yeah.
You're using citations as your marker, but we're notoriously databases are undercited. They'll stay in the text that they got the information from Uniprot or Intact or wherever, but they don't cite us.
Yeah.
Is there any way of compensating?
No. So we're hugely undersampling with all of this, and so another piece we will add is mentions of the names of databases, and in the case of it's already true for Uniprot, but it's certainly true for sequence databases and many others that often authors don't say. They say we use this sequence or we use this protein record, and they just give an accession, and because it's so established, the editors know how to mark it up and put in a hyperlink. Everyone knows what it means, but it's very difficult to find. And of course, most people, I mean, there's an appeal to using citations of accessions because a lot of people will come to a data resource, they'll come to a model, they'll come to Uniprot or something, and they'll look for something, and they'll do a search, and they'll have a load of results, and say, oh yeah, that's interesting, I don't really care about that, and they'll find something. They'll actually have looked at and possibly even retrieved quite a lot of things, but the things they actually used in a significant, substantial, they used everything, but the things that really meant something for their study, for their onward study, would be a subset of that. And citation is nice in a way because what it does, you don't cite a data record. You have to go out of your way to cite it. So you don't cite it unless you really, I think, unless you really mean it, or unless the editor has thought, yeah, you really should be citing that record here. So it's never going to be, it's always going to undersample hugely. And it's not the only way of doing this, but it's already, it's already indicative of huge use, which is a very positive thing.
So time is up. Shall we?
Oh, it's one. Oh.
Can I? Sorry.
Please.
I was actually wondering about whether or not the database that you're building is going to be accessible and mineable by other people who are like not part of the core group.
Absolutely. It's not yet, just for practical reasons. We're just still setting it up. We're not planning on running this as any kind of a service. So it will be data sets in a structured way. For the inventory, so the discovery from the literature of the long tail of the 3,000, the 4,000 data resources, that's already published, and we actually use the services of a consultant to check. So we publish the scientific part of the results, and we publish the methodology as an open workflow with all the data. And we actually made a point of getting someone in to check that they could actually recreate it. So we very purposely made this open. We're a very open organization, or aspire to being one.
So let's thank Dr. Guy again. And